This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 685, recorded on November 25th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It's a nice sunny day, 52 Fahrenheit, 11 Celsius.
And from Austin, Texas, Rich Condit.
Hi there, 58 degrees, sunny. The website says it's cloudy, but the weatherman is not looking out the window. It's sunny, and it's gorgeous.
And from southeastern Michigan, Kathy Spindler.
Hi, everybody. I have to be Debbie Downer about the weather. It's 43, rainy, and cloudy. Feels like 37, and that's 8 degrees Celsius.
Apparently, a lot of people are traveling, even though they shouldn't be. Oh, well.
Yep.
All right, we have a bit of news. We have two papers for you today. Bit of news. We'll try and be concise, but I'm not sure it's in our DNA. As many of you know, Monday morning at 6:54 a.m. This seems to be the time when the press releases go out. Oxford AstraZeneca vaccine efficacy. And, I have been told that I try and use the right words. I do, like mutation referring to changes in nucleic acids. Efficacy is the right word here, not effectiveness. Effectiveness is something else. Efficacy is when you do a vaccine trial and you measure how well it works. And so this is a very interesting situation. Of course, it's a press release. So this is the chimpanzee adenovirus vector, which is carrying the gene for the SARS-CoV-2 spike glycoprotein. It developed at the University of Oxford and partnering with AstraZeneca. And this is, of course, being phase 3 tested in many people, tens of thousands of people.
It's over 30,000, I think. I had to look it up, actually.
So this is an interim analysis, of course.
Yeah. And this is the interim analysis of the UK and Brazil patients.
Right.
The US part of the trial was paused for a month. And so they are behind. Yeah. And so there is no US data here.
They're still enrolling, in fact.
So overall, 70.4% efficacy, which of course is lower than the two mRNAs, which are in the 90s. But they did two regimens, and they had 90% in one regimen and 62% in the other regimen. And you may ask, well, what is this two regimens? And many people wrote in to send us this story. So one regimen, 90%, and the other 62%. Apparently, for a number of people, they only gave half a dose for the first inoculation rather than a full dose. So some people got a half dose, then a full dose in the boost, and others got a full dose in the first and second shots. And when you do the full dose in the first and second, you get 62% efficacy. And when you do a half dose followed by a full dose, which was apparently a mistake, you get better efficacy. And I will let Kathy explain the basis for that. She is the adenovirologist.
Well, it's interesting because several of the articles have different speculation as to why. Mm-hmm. But only one did I find the speculation that it could be that the one where there were full doses given both times, the first full dose of the adenovirus-based vector elicited a fair amount of antibodies to that vector. And so that with the second dose, then the efficacy was reduced because the antibodies got rid of the vaccine vector.
Yeah.
And that by giving a lower dose the first time, less of that happened.
So that's theoretically, they didn't actually measure anti-vector antibodies. Okay.
Totally theoretical.
Right. I've seen kind of three different potential explanations. That's one, and I think that one makes a lot of sense and it has a lot. One of them is really more of a numbers and statistics detail, because this was done in, a smaller group of the patients. And so some people said, oh, it's just statistics. The other thing that we do know from some vaccine studies is that if you do what are known as prime boost studies, where you give, especially say DNA for the prime for the first shot and an adenovirus vaccine for the second or something like that, there are definitely papers that show that all primes are not the same. And different types of primes will give you better types of immune responses on that boost. And so there may be differences in how those memory cells are first generated. That hasn't been shown necessarily for an adenovirus like this, but that is known to be the case with making other vaccines, is that different boosts can set up that immune response differently. So the idea that a lower dose on the prime could be beneficial isn't totally shocking, but I do agree with Kathy that the explanation that made the most sense to me had to do with anti-adenovirus vector immunity.
Brianne, in the situation you're talking about, do any of these studies involve a prime and a boost with the same antigen?
They almost all are the same antigen,
except the Russian.
with different
Remember,
modalities.
the Russian vaccine has 2 different adenoviruses.
Yes.
So that takes care of the problem, because it is an issue if you're going to do 2 shots to have a vector. So the mRNAs don't have this problem, obviously, because there's just lipid.
There's been an issue in not only the vaccines, but in gene therapy vectors since the get-go.
That's right.
Any sort of viral vector thing, this is potentially an issue. And that's why they use the chimp adeno as a vector, because at least in the first dose, people don't have antibodies to the chimp adenovector. And that's why in the case of Ad5, which is another vector that's used in vaccines, there is a concern, because that's a pretty common human adenovirus. And that's why Johnson & Johnson chose to use adeno 26. I think one of the reasons they chose to use it is because that's has a much lower seroprevalence in the human population. By the way, the trials are, the UK trial is 12,390 participants, and the Brazil trial is 5,000 participants. Okay, so the trial sizes. This is the other thing, is that if I'm not mistaken, this funny half-dose prime thing was in the Brazil trial. Okay, I think that's correct. So it's a smaller population, but not by much.
Yeah, and
at least in terms of the overall study size, and it's a different population.
Yeah.
I don't know how different. There's Brazil versus the UK, but that's another. So
all of the Brazilian participants got half dose, or just some of them?
I do not know.
So can't possibly be if you said it's 5,000 in Brazil, because it's supposedly fewer than 2,800 got the half dose.
So I suppose
but those numbers that you cited, Rich, don't add up to the reported 23,000 participants whose results were reported Monday.
Right. I'm reading off of the clinicaltrials.gov that are cited in the Times article that we read, but there's other AstraZeneca trials out there. Okay, so the data may incorporate some of the other trials as well.
Right. Today's Times article mentions 23,000. So it's 2,800 got the half dose out of the 23,000. So different population, could be different age, etc.
So because it wasn't, an original plan, they probably didn't think about whether the people who were getting that dose were randomized and were.
That's right. When you make a mistake,
a completely random population.
Yeah, when you make a mistake, you don't randomize it, right? So 60% is not terribly good. I mean, it would get licensed, but that leaves a lot of people unprotected. So the question is this a problem for any ad-vectored or virus-vectored approach where it's the same vector in the prime and boost overall? I would think so, right?
I don't know. We'll find out.
Potentially.
And one of the others, I think the Johnson & Johnson, they don't do a prime and a boost.
Right. I think Johnson & Johnson is just one dose.
That's right. That's right. We'll see if that gives a good efficacy. Now, the other question is, what is AstraZeneca Oxford going to do? Are they going to do half full or full? Is the glass half full or half empty?
Because the U.S. trial is still enrolling, and they have released the details of that trial as opposed to the UK and Brazil trial. My quick reading of this made me think that they might try the half dose in the U.S., but I could be wrong about that. Can't find that.
There's certainly motivation to do that.
Yeah,
because
for
if
sure.
this difference is real, it's
I mean,
huge.
if you can pivot, to use a word that Sally used yesterday on Immune, right? Pivot.
Pivot.
If you can pivot quickly in the middle of a trial, that would be good because...
I did find it here. It says AstraZeneca plans to start testing the half-strength initial dose in its continuing U.S. trial.
Okay.
And asked the FDA for guidance on how to proceed.
Good. Now, the other issue is a number of people have asked, based on this mistake, why should I have confidence in these vaccines? And I don't know what to say to that, folks.
So I would say, one, have you ever made a mistake? Two, when you do you fess up and say, I made a mistake?
And do you fess up to the entire world in the New York Times?
Right. And then do you move forward, capitalizing on your mistake?
Yeah. Did you learn
and do the right thing? You know?
I think he is a mistake, which may be informative, right?
Yeah.
I mean, Alexander Fleming.
Nobody's been hurt.
Alexander Fleming's mistake got us penicillin, right? Many examples.
Nobody's been hurt. Nobody died. If anything, people have benefited.
Yeah.
Nobody has, this has not been FDA or otherwise approved yet, okay, because they haven't seen all the data. They uncovered this in the early stages. So everything's fine.
I think the fact that they picked it up is important, right?
Yeah, and to me, the fact that we are having this conversation is related to the fact that while they did have this mistake in dosing patients, they caught it and recorded it. Yep. We are able to actually look at the data with the patients who got the different doses.
Yeah.
And so, there was a fair amount of care that went into this and recording of what was done, which is important.
One problem here is that we live in a, both locally and globally to some extent, in a culture of deceit, okay, where you don't, where you make up your mind and you can't change your mind, or you're waffling, okay, or you exaggerate or stretch things or outright lie to get your point across, and that's not the culture of science, okay? The culture of science, by and large, trust science, not the scientists. People do screw up now and then. Okay, there are evildoers even in science, but by and large, the culture of science is truth, okay, and getting it right.
But although
I can think of two examples. One is the Cutter incident, from which we learned a lot about making sure that when you scale things up, you test the process again. That goes back to the 1950s. And so you can be sure that everybody giving any kind of vaccine is going to be double, triple, quadruple checking their doses and their calculations. And then just thinking back to, some NASA screw up where something was measured, or was supposed to be metric and it was standard, or vice versa. I don't remember the details, but we learn from those mistakes big time. So I think that's what's happening here.
I can imagine, though, some governments where this would not be revealed, right? It would be hidden.
Oh, yeah. Good point.
And I think that's happened in the past, and will happen. I'm not indicting anyone, but it could happen. But yes, I think a lot of people will not appreciate this and will say, I'm not getting that vaccine, or maybe any vaccine, but I think I agree with the sentiments. Kathy, you have some comments on costs here?
Oh, yeah, that the cost of this AstraZeneca vaccine is considerably less, maybe about, now I've seen $3 to $4. The first thing that I saw was that it was, I think when subsidized, somewhere on the order of $2.50 a dose compared to $15 to $20. I'm not finding it in the show notes, but yeah, there's a substantial difference in the cost. And of course, this adenovirus vaccine doesn't have the cold chain issues that the mRNA vaccines will have.
So remind me, what are the storage conditions for the chimp adeno vaccine?
I don't know, Rich.
My understanding is that they want you to ship it and store it refrigerated.
Refrigerated, and it will last 6 months at
so it
2 to
doesn't have to
8.
be frozen.
2 to 8
That's
degrees
what I thought,
Celsius,
but
6
I figured
months.
someone else would know better than I would.
It's in this press release thing
2
here.
to 8 degrees Celsius, 36 to 46 Fahrenheit.
Yeah,
Vincent, you're cutting out again frequently.
It's probably on your end because
there you go.
Everything's good here. I shut off my Dropbox, shut off everything except what we're doing. Pfizer's vaccine $20, Moderna's $15 to $25, and $250, as Kathy said, for the adenovirus, AstraZeneca. We have 2 papers for you. One is very short, and it is less than a page. It is an addendum, so I guess it's not really a paper. It's an addendum, right? Corrections and amendments. Sometimes these have to be reviewed because I've handled some of these before. I don't know what Nature's policy is, but it's called Addendum, a pneumonia outbreak associated with a new coronavirus of probable bat origin. So that is a paper published back in February, and it was submitted on 20th January from Zhongli Shi's laboratory in Wuhan. And it was the first report of this, the characterization of SARS-CoV-2, where they had isolated virus from bronchoalveolar lavage fluid of patients with pneumonia. They did the genome sequence and they lined it up and found that it had high homology with the highest nucleotide sequence identity, the highest nucleotide sequence identity with the bat coronavirus called RATG13. So this is an addendum to that paper. And they say we provide further information about RATG13. So I think one of the issues here is that they changed the nomenclature of RATG13 at some point after its initial isolation in 2013. And it wasn't actually a virus isolation. It was a fragment of RNA polymerases, as I'll tell you in a moment. And people have said, you changed the name, so you must be doing something nefarious. So this is in part an attempt to clarify exactly what they did. So here's the story. So this happened between July and October in 2012. They got 13 serum samples from 4 patients who had severe respiratory disease. And these patients had visited a mine cave in Yunnan Province, China, which is where they have lots of these caves and they're full of bats. They had gone in the cave not to, have lunch or anything, but they were cleaning bat feces out of the cave because they wanted to mine copper. All right, so they're cleaning the feces out of the cave, and we know that bats shed viruses in their feces.
This just blows my mind. We have wondered what people are doing in these caves in the past, and this is I can't imagine a heavier duty exposure.
And remember,
some of these caves,
they're probably not wearing face masks, right?
Oh yeah.
Right.
They're just in there cleaning stuff out.
Whereas when Zhongli Shi's group goes in to collect, they're probably all gowned up, right?
Yeah.
We just recently watched with the grandkids. We've been going through the old Planet Earth series, and they've got one episode on caves. And I forget where this cave is, but it's a bat cave where they have a mound of bat guano that's like 100 meters high. All right, crawling with cockroaches. Okay.
Oh my gosh.
And so I can imagine that a lot of these places cleaning out the bat guano. I mean, the exposure is just phenomenal.
Cleaning out the bat guano.
And I also want to point out that this is 2012.
Yes.
Okay, when this is happening, and they got people getting sick, and they're wondering whether they're infected with some pathogen from the bats, and they're doing the right experiments.
Okay, so they got samples from these patients, and because they were hospitalized, they got sick shortly after doing the cave cleaning. They tested them they did PCR using primers for the RNA-dependent RNA polymerase, Ebola virus, Nipah virus, and a bat SARS-related coronavirus, RP3. And all of them were negative for the presence of those viruses. They then took serum samples. They looked for antibodies against the nucleocapsid protein of these 3 viruses. None of them were positive. And they say recently we retested these samples with an ELISA against the nucleocapsid of SARS-CoV-2, and no reactivity. So these patients were not infected by SARS-CoV-2. And I think, so this paper has gotten a lot of coverage in the press, and these articles say this vindicates, in Zhongli Shi's words, this vindicates, and it shows that this virus didn't come from their laboratory. And I think the fact that these patients were not infected with SARS-CoV-2 is part of that. So, but what were they infected with? So they did a lot of sampling in these caves and of various animals, bats, rats, and musk shrews. They found alpha coronavirus and paramyxoviruses. But between 2012 and 2015, their group sampled bats once or twice a year in this same cave and collected 1,322 samples. And in those samples, they detected 293 highly diverse coronaviruses. 284 were alpha coronaviruses. 9 were beta coronaviruses. This is on the basis of RNA polymerase sequences. And all 9 beta coronaviruses were SARS-related coronaviruses. And one of this was given the sample ID 4991. That's the one that was renamed RATG13 in the article. And they say we renamed it to reflect the species of bat, the location, and the sampling year.
And in that article is still a partial sequence. Is that correct? I don't think they put the whole thing together until the outbreak.
In January, they went back. So in January 2013, in the other paper, of which this is an addendum, they said the highest percent identity of SARS-CoV-2 is with this ID 4991, and they had just the RNA polymerase sequence. So they went back in the freezer and sequenced the whole genome, and then they renamed it. Right. And I, for this, in preparation for this, I reread that February paper, and it's actually pretty remarkable. Yeah, it's a great paper. It's coming very quickly during the outbreak, in the very initial stages of the outbreak. It's remarkably thorough and insightful. It's very good. So for our listeners who, don't think a ton about viruses, or who haven't spent as much time thinking about viruses, to me, there's one other piece of the text that Vincent talked about that stands out that I really want to point out. So between 2012 and 2015, the group sampled bats in this one cave in one place in China. They have over 1,000 samples, and they detected 293 coronaviruses, by which we kind of mean they found 293 viruses they'd never found before, that had never been previously known to the world, looking in one cave over 3 years. And so I think that people sometimes don't realize how many viruses we haven't discovered and we haven't named. And so they think it's shocking that there could possibly be a new virus. Yeah. But here in one cave, they found 293 new viruses from this one group, the coronaviruses. Yeah, and there were like 6 species of bats in this cave. About half the bats were coronavirus positive for one virus or another. So that's half the bats, okay? They found the same coronavirus in different species of bats.
So some of these coronaviruses can cross bat species, and they found a high fraction of these bats had more than one different type of coronavirus, which is relevant because coronaviruses can recombine with each other. And in fact, SARS-CoV-2 is thought to be a recombinant of a couple of other coronaviruses from earlier on. So just to reemphasize what Brianne said, this is a zoo, and it's a real melting pot. And this is one cave.
Someone wrote to me kind of out of the blue. I didn't know them this week, asking me to comment on something that they had seen where Bill Gates was talking about the next pandemic. And I think they were sort of thinking this was nefarious and that, Bill Gates knew something. And I just pointed out, all he knows is what we all know, is that pandemics keep coming. There are lots of viruses out there. It's a foregone conclusion that there's going to be more of this. And we need to have pandemic preparedness as a result.
This, so again, to point out this sample that they originally called ID 4991, that was published in 2016. It's in an article in Virology Sinica, coexistence of multiple coronaviruses in several bat colonies in an abandoned mine shaft. So this is part of the sampling that they're talking about in this addendum. And it was named 4991 in that paper, renamed in the 2020 February Nature publication, RATG 13. And I remember speaking with a reporter back in March who said, don't you think it's strange that they renamed this virus? Doesn't that suggest to you something nefarious? I said, no, it doesn't suggest anything nefarious. We rename things all the time. Come on.
And the fact that we know that they renamed it means that they're not trying to hide something.
Yes. Now here they're telling us why they renamed it. Well, we called it to reflect the bat species, the location, and the sampling year, 13. So
yeah, it's pretty clear that they went from a non-descriptive name to a descriptive name.
Yeah.
I wonder what
in particular, since it had acquired significance.
Yes.
Okay. As the closest relative to SARS-CoV-2. By the way, they called SARS-CoV-2, what was it? SARS-NCoV. Mm-hmm. Okay. That got renamed.
Yes. It was 2019-NCoV. That was what WHO had originally called it. Yeah. So I wonder what those people were infected with. Obviously, that gave them the pneumonia, though. We don't know. And it seems to me that if they had, lavage samples from them, they could sequence them and find out. But I haven't mentioned it.
Yeah, back then, I'm trying to think. So the sampling was done as early as what was between 2012 and 2015. The paper was published in 2016. Metagenomics has come a long way since then. So it could be that if those samples are still around, you could find out what was there.
Yeah, so what they done, they're just doing PCR with primers to the RNA polymerase, right?
Very targeted.
And very targeted.
To a limited number.
And if this homology is not, if the identity is not high, you're not going to amplify anything. So, okay. Fascinating. So many, and another thing, just to go back to Brianne's point, I remember people ask me, how could there be a new virus? Are you kidding? There's so many out there.
Exactly.
We got plenty out there.
Yeah, and importantly, it's not necessarily new.
No.
It's new to us.
New to us.
New to us. Now, actually, SARS-CoV-2, we think, is new in the sense that the best data say that, it resulted from some recombinational mix-up of a few bat viruses around in the 60s sometime, right?
Yeah.
So
it depends what you mean by new,
if
right?
That's new. That's new. But
I mean something related
these
to
things
it
are out there.
Something related to it was clearly circulating in bats. It had to be before it gets into people. Now it may have changed a bit in the earliest people, right? But it would still be SARS-CoV-2, right? So, I mean,
the planet is crawling with viruses, folks.
It is. And so are all of us as we sit here.
Yeah.
Mm-hmm.
We're crawling.
I told people on my cousin's Zoom about viruses being on cabbage, and one of them said, "Oh, that makes me feel not so good.
Yeah. So I always,
They've been there
I
all
always
along. They're
describe
insect
that.
viruses. Don't worry about them.
Okay, now we have a very cool paper, which, one could argue that we should do on immune, but since we do two episodes of TWiV a week, it's quicker to get to it. I think it's very important, and it goes back to a paper we discussed a few weeks ago. So the title is, it's a journal pre-proof. It's going to come out in Immunity, and the title is SARS-CoV-2 mRNA vaccines foster potent antigen-specific germinal center responses associated with neutralizing antibody generation. This is from a number of groups. So the first author is Caitlin Lederer. The last author is Michaela Locci. It's from the University of Pennsylvania, the Universidad de Antioquia in Colombia, Duke University, Mount Sinai, Aquitas Therapeutics in Vancouver, and then Duke again. Okay.
The first two authors are co-authors, Caitlin Lederer and Diana Castaño.
I bet Diana Castaño is from Colombia. Actually, she's from 123, which is Penn.
One of which is Colombia.
One of which is Colombia. I show I gave a talk yesterday to some biotechnology students in Mexico. They wanted to know my history. So I show a paper I published with David Baltimore with just me and David as co-authors. The old days when you used to publish a paper with one other paper. I'm sure that Kathy and Rich have papers with one other person. Maybe you too, Brianne. Do you have papers? Because you're kind of young.
I don't think so. No, I don't think so. I think even when I think of a review, I think there are three of us.
That's not bad. Three is, but nowadays, all these, it's fine.
I think I even have a sole author paper from graduate school.
Whoa.
I have to look back and be sure of that.
I think you have mentioned that. Yeah.
My first paper here when I moved to Columbia was a sole author paper. It was experiments I had mainly done here on my own when I first got here. I thought I could do experiments for a while. I actually did. Anyway, so this is cool. All right. So, for a vaccine to be effective for a long time, you need to have long-lived plasma cells and memory B cells. And the formation of those depend on germinal centers. Now, Brianne, of course, jump in anytime and correct me. Germinal centers.
I'm a big fan of this paper, so I'm excited to talk about it.
And germinal centers we talked about before. I don't know who was on the TWiV, but we did a paper where they showed in patients with severe COVID-19, people who died, basically, their germinal centers are trashed, so they can't make long-lived antibodies because the long-lived antibodies come from these very cool affinity maturation reactions that happen in the germinal centers where you have, mutation of the antibody genes and multiple rounds of selection for high affinity. And those are the
so I think we talked about that paper on immune, actually.
Yeah.
Is that right?
Yeah, yes. I think so.
Okay, I'm confusing my podcasts. Oh my gosh.
But I would definitely recommend, if people are interested, that people look at that paper because the first figure in that paper shows microscopy images of germinal centers from patients who were infected with severe SARS-CoV-2 infections, as well as healthy donors.
Yeah,
and there's a dramatic difference in germinal centers. I'm planning on having it in my germinal center lecture because it shows them so nicely.
You do a lecture only on germinal centers? Mm-hmm. You do one lecture on germinal centers?
I don't do one lecture only on germinal centers, but there's one lecture where germinal centers are take up quite a bit of time.
So are the differences both qualitative and quantitative?
I would say yes, although it's the reason why I hesitate is because the germinal centers are almost completely destroyed. So there are certainly fewer of them, but they also are just look incredibly different. And as they go through different types of staining, there are different types of cells present or absent in some of those areas. And this is important, as Vincent said, because the germinal center is a location where B cells that have seen antibody are able to differentiate to either make good memory cells. So you're going to get a good memory response, a long-lasting memory response to the vaccine, as well as making really good antibodies that can bind tightly to the antigen. And so to make your best possible B cell response, you need a germinal center response, and you need a particular type of T cell in that germinal center called a follicular helper T cell that can help out those B cells in order to make their best possible response.
Now, Brianne, you've just done some of this, but since that paper was done on immune.
Yeah, and you don't listen to immune, right?
And I don't listen to immune. Actually, I'm going to make another. Let me reference, I'll do it now. TWiV 161, Concerto in B.
Yes.
With Gabriel Vittora from 2011 was all about germinal centers. And I would refer listeners back to that. That's the hardcore, that's more than you ever wanted to know about this. But there's probably a lot that's happened since then. Brianne, could you give us a primer on germinal centers? Where are they? How do they arise? What do they do? Do they stick around? Blah, blah, blah.
So germinal centers are areas in either lymph nodes or spleen, really in any secondary lymphoid organ, where B cells and T cells are gathered together after we have started to make an adaptive immune response. And that is where the B cells are going to differentiate into making memory cells that can live for a long time, give you a long-lived response to the particular antigen, as well as differentiating into plasma cells that can secrete high levels of antibody. And we see this process of affinity maturation, where the antibodies made by a particular B cell get some tighter binding to antigen, along with a few other processes in B cells. And so these are generated after infection, as your B cells are starting to really get ramped up in the lymph node.
So when I get my mRNA vaccine injected into my muscle,
yes,
presumably at some point, the antigen, the spike antigen, is going to be expressed in an antigen presenting cell like a dendritic cell, right?
Yes.
And that dude then circulates and winds up in a lymph node, right?
So that dendritic cell will move to a nearby lymph node. It's called the draining lymph node for whatever region of the body we're talking about. So it will move.
Vincent just pointed to his armpit.
Yes.
For those of you who are just listening.
Yes, which would be the draining lymph node if you were getting a vaccine in your arm. So the dendritic cells will move to that draining lymph node. They will start activating those T cells. And once the T cells are activated, they will interact with the B cells in the germinal center.
Okay. And when the germinal center is, there's a lot of cellular proliferation going on.
There is.
And there are specialized regions in the germinal center. So this is why when I go to the doctor and he wants to know if I've had an infection, at least when I was a kid, now he takes blood and stuff. He palpates my lymph nodes. Is that right?
Exactly.
Because if they're big, it's because my germinal centers are growing.
Yes, because
right?
your B cells are dividing and dividing and dividing and dividing and dividing, and there's so many of them that they are making your lymph nodes awfully crowded.
And
Now,
Rich,
if I establish a germinal center,
what is the name for the swollen lymph node, Rich? The medical name for it.
I don't know.
Think HIV/AIDS patient. Yeah, there you go.
Oh, jeez. Where did that come from? I got to teach Porter that one. Lymphadenopathy. That's cool. So can the news spread from one lymph node to another? Or do I have to seed germinal centers in multiple lymph nodes from the original site of immunization or from independently?
So you can see some movement from one germinal center to another. Actually, remembering back to that particular paper, they showed both the draining lymph node and the spleen and showed the same things going on in both of them with germinal centers. So there is some movement from lymph node to lymph node, although the B cells and T cells, when they get activated, have a little bit of knowledge of, I'm getting activated in a germinal center near the lungs. Maybe I should go be patrolling the lung for my antigen later on.
I have a question about the the T cells that are getting activated. Are those just sort of garden variety CD4s, or are they specifically the T follicular helper cells, which I'm assuming must also be CD4 positive?
Yes. So anything that's helper is CD4 positive. So T follicular helper is going to be a CD4 positive. Really, what happens with that CD4 T cell when it's getting activated is it's a garden variety T cell.
Okay.
And the particular cytokines that dendritic cell is making, the particular response that dendritic cell is making, will tell the T cell which subtype of T cell to become.
Okay.
So it might say, I have an antigen, and this antigen is from a virus. So please grow up to be a T cell that responds well to viruses.
Okay.
Or something like that.
So, and once the infection is cleared, and this is all over, those germinal centers then decay, if you like. They more or less go away, but I'm left with long-lived plasma cells and memory B cells.
Exactly.
So I'm pumped for the next round. Now, when I get boosted or reinfected, do I go through this process again and mature even better?
Yes, you do.
Okay.
Some of those memory B cells will go back to a germinal center and will...
They'll go to graduate school.
They'll go to graduate school, absolutely. Further differentiate.
This is so cool. This just blows my mind.
They go to graduate school. They go to graduate school, not medical school.
Okay. Do I have the ability to share my screen?
Yeah, I think so. If not, I will give it to you. Try it.
All right. Let's see here.
While you're doing that, the thing about the paper we did on immune that's cool is that in these patients whose germinal centers were trashed, they make antibody, but it's terrible. It's low affinity and there's no memory cells produced. So that just kind of, and this is from that paper, right, Brianne?
Yeah, so this is just one of the images from that paper. This is figure 1, if you're looking at B. The 3 images that are right here on the left are from a severe COVID-19 patient. Those are the germinal centers and the draining lymph node. These are what germinal centers look like in a healthy individual on the right. And so you can see how dramatic this difference is.
So, of course, I haven't listened to that particular episode of Immune, of course. So is there a mechanism? Do we know why they're trashed?
There is no mechanism. I have been speculating on that myself quite a bit.
They actually in the paper speculated on IL. What was the IL that they speculated? Because there's some mouse model where they they did therapy with that and it restored germinal center formation. Remember?
So that was with, I'm trying to both undo my screen share and remember how to answer that question.
But anyway, there was one in mouse models. There was one cytokine implicated, and I think there has been some trials in people for other reasons that, and this has been shown to help germinal center formation. So yeah, there's some hints. Yeah. Okay. Now, so this paper is important because they want to know in some of the two different kinds of vaccines, mRNA vaccines and a purified protein vaccine. Do they make good germinal centers? Do they make good memory B cells and long-lived plasma cells? It's a really important question, right? Because if you have a choice, you'd like to know which is better. And there is a winner here. There's a very clear winner.
Yeah, and the other thing that's sort of interesting with that is typically when people think about a protein subunit vaccine, they think about a vaccine that is really kind of driving antibodies. A protein vaccine is thought of as kind of like the antibody maker.
Yeah.
And the nucleic acid-based vaccines are thought of as making some good T cell responses, some good antibody responses, being a little bit broader. And so that was one of the things that really, impressed me about this paper, was testing that immunologist lore.
Yeah, and we'll see more of this hopefully as vaccine trials proceed. It's important. So they have now. None of these people on this paper are from Moderna or Pfizer. So I think they're making their own vaccines, an mRNA vaccine.
I looked at the methods. They're making their own vaccines.
So they make an mRNA spike encoding vaccine. It's a pre-fusion version, so it will not be cleaved and reorient. So you get all the right epitopes that are needed to neutralize infectivity. And then they have a protein-based vaccine, which is just a receptor binding domain, right, with an adjuvant.
Right. And I think it's significant that it's just a receptor binding domain. So it's only a fraction of the spike protein. It's not the whole thing. It's the active portion of the spike protein, but it's not the whole thing. And there's no, as far as I could see, there's no attempt made to make any kind of a nanoparticle or anything. This is just going to be free soluble protein. And I don't think that there's any, there's no structural data. You don't know kind of what the conformation of this is. So not all, I want to make the point that not all protein vaccines are the same. There's a protein vaccine in trials that's in fact full-length spike that's arranged in a nanoparticle. It would be interesting to see what that does in this context, because it may behave entirely differently.
That's Novavax, right?
Yes.
Okay.
Right. So this paper is sort of comparing apples to wrenches, and maybe if they compared it to Novavax, it would be apples to oranges.
Yeah, maybe so.
Apples to wrenches. Wow. What kind of wrench?
Alan.
Oh, of course.
I got one right here. Look, my desk.
Yeah, there you go.
Why do I have an Allen wrench on my desk, you may ask? I got all kinds of tools on my desk because I'm always fooling around. Anyway, sorry. I promised I wouldn't wander. Anyway, so they do these experiments in mice, which are great because you can, manipulate them extensively. You have antibodies to stain the different kinds of cells you're interested in, and you can do flow cytometry. But the downside, of course, is they're mice. They're not people. And as you know, mice lie and monkeys exaggerate. You do know that, right? So,
well, if they've been listening long enough,
they know
that.
So
it doesn't hurt to remind them, mice lie and monkeys exaggerate.
And so
it's also worth pointing out that the protein vaccine is delivered with an adjuvant,
right?
Okay, which is they describe it as an oil water emulsion. Mm-hmm. Okay, and that, as I've described it before, is supposed to serve to sort of piss off or wake up the immune system. Okay, and help out with stimulating an immune response.
This particular adjuvant is called Adenovirus.
Right.
Yeah.
Whereas the messenger RNA vaccines, they're essentially self-adjuvanted or don't need to be adjuvanted. They've got the lipid nanoparticle, they've got kooky stuff in the RNA itself, and they're making protein on site. I think all of those have potentially adjuvanting effects. Am I making sense?
Yeah.
All of that is good. There are lots of ways that nucleic acid can piss off your immune system just by itself. That would be self-adjuvanting.
So the first experiment, they immunized mice with either the RNA or the protein vaccine. They wait 7 days. They look for germinal center B cells. After a single immunization, no germinal center B cells induced by the protein vaccine. And in the mRNA vaccine, they say a robust population, which is a qualitative statement, but they got them. They got germinal center B cells. And they also can find short-lived plasma cells in the mRNA vaccines. And they looked 14 days later. And there, the protein induced minimal germinal center responses. And they say a sizable germinal center B cell population in the RNA immunized animals.
The data are striking.
Very striking.
There's just no question about it.
Right.
They also
and the
look
pictures of the lymph nodes with and without the germinal centers are striking.
Yes.
Yeah, they have microscopy where they can look at these lymph nodes from mice because you can do that. You can take them out and you can see the germinal centers. You know, they have antibodies to stain particular cells that are part of the germinal centers, and you can see follicular dendritic cells, IgD positive, naive B cells. So, and they see these nicely in the mice that got the mRNA, not the protein. I mean, just there, it's amazing.
Yeah, it's very striking.
Yeah, and I think that this is really important for a couple reasons. One, after looking at the paper showing that lymph nodes are trashed in SARS-CoV-2 infection, sometimes people said, oh, that means that vaccines are never going to be possible. You can't make long-lived memory to these antigens. And here they're showing no, with a vaccine, it may, there does seem to be some possibility. We're getting some germinal centers here, and that's good. And that is great for vaccines. And it also might give us a little bit of a hint with Rich's question about mechanism. Perhaps one of the not spike proteins is messing with the immune system. And that was what was happening in the trash germinal centers. And when you give a vaccine with just spike, you don't have that effect.
Going back to this thing that we were talking about, whenever it was, last Friday or something with Shane, and that is that, and this never ceases to fascinate me, is that a vaccine doesn't necessarily have to precisely mimic an infection or the immune response to an infection. And in fact, it's possible, I think quite possible, to make a vaccine that does better than natural immunity by basically fooling your immune system.
Yep, absolutely.
So they also look to see, so, okay, we get nice germinal center formation, but is it antigen specific? Is it specific to SARS-CoV-2 spike? And they look at that. They make fluorescently labeled protein basically, and they can do flow cytometry to look at whether it's antigen specific. And they find that the mRNA induces antigen specific B cells, but not the protein vaccine at 7 days after immunization.
I want to remind, I haven't done this for a long time. I want to remind the new listeners just in just a couple of sentences what flow cytometry is, because it's a miracle. Okay. You take a suspension of cells, you label them with fluorescent labels of one sort or another that are specific for various characteristics you want to see. And this machine actually examines individual cells one at a time and tells you what their staining characteristics are. Okay. And then keeps track of the numbers of cells with each of those characteristics. And if you really want to be cool, you can actually tell the machine to sort those different cells into different pots. Okay.
You have to have the fancy machine for that, though.
Okay.
Brianne has
a little machine won't do that.
Brianne has a little one in her lab like this big. It's amazing. How many colors can that do?
The one in my lab does 4 colors and 2 physical parameters.
And the cool machines?
The ones I used in grad school were 16 colors, and there are machines that do a lot more than that.
So that was in the Letvin lab?
In the Letvin lab, we did 16 colors.
Now you get to colors in quotes,
Yeah.
right? These are different fluorescent wavelengths, I would assume.
Exactly. Well, part of the reason why I can't give you a current answer is that there are some cytometers now that are combined with mass spec.
Yeah.
Ooh.
So that you can get 50 plus parameters, but then it's really not colors.
It's awesome.
And how much money are we talking for, say, the machine?
I have no idea how much a cytoph costs.
But, this is a
Not
problem
cheap.
because then these immunologists go crazy and they define all these new cell populations based on It's just
Be
out of
different
control.
parameters.
As someone who likes flow cytometry a lot and who has done a lot of flow cytometry in my life, I would really like to point out that I am not the one who called it a miracle. It was Rich.
I have a question about a highly technical term, which I noticed in this paper. I don't think I've seen it before. When you stain with these different antibodies to identify different populations, then it's called gating and so forth. But they talk about a dump minus population.
Yep. So what you do in that situation is sometimes you want to look at B cells. And so you want to you might pick a protein that's on the surface of B cells to examine. But the alternative thing that you can do is you might take a protein that's on the surface of T cells, and you might take a protein that's on the surface of NK cells. You kind of get all of the things that are not B cells. You make them all the same color, and then you say, I want to exclude anything that binds to something that color. I'm getting rid of my T cells and my NK cells and my anything else. And you call that color or that channel in your analysis the dump channel. So anything that you have identified as a non-B cell, you're getting rid of by labeling it with the dump.
Nice choice of words.
Start listening to Immune.
Yeah.
Nice choice. Well, the one yesterday with Sally was great, but the one where we did this paper is also good as well. Then they look at the kinetics of formation of these germinal center B cells, and they see that, if you look up to 21 days, you can see some coming up with the protein, but much more much sooner and higher in the mRNA animals, immunized animals. Then the next question is, are there memory B cells made? Which, that's dependent on germinal center formation. So you can almost predict if there's no germinal centers, you're not going to have memory B cells, and they can look for these memory B cells again by their flow cytometry. And you get nice memory B cells with the mRNA inoculation and not with the protein. And that's a summary, but if anyone wants to embellish, they're welcome to. Sorry, go ahead.
I was just going to say, again, to me, one of the big surprises here was that when I think of a protein-based vaccine, I think of antibodies. And so to see that the mRNA-based vaccines are making kind of better memory B cell, potentially a better, antibody response was really impressive and eye-opening to me.
I told someone the other day, I was brought up in RNA labs, and I was brought up to never look sideways at RNA, otherwise it would degrade, And the idea that you would inject RNA into someone is, I always thought, are you crazy? It's all going to degrade. But obviously they have protected it beautifully. And so...
it is pretty amazing because the concept of DNA vaccines has been around for a long time. And it's not nearly as successful for reasons that we could talk about another time. But it's amazing it's taken us so long to get to RNA vaccines. It's probably for a variety of reasons, that being part of it. Okay. But these are stabilized with funny nucleotides, and then the delivery involves nanoparticles.
But
and, there's a lot of technology.
RNA rules. Remember, the RNA world was first on Earth.
I'm looking at Kathy here. Before
a DNA world.
I'll have to find the cartoon that somebody sent me yesterday and show that to you guys after the show about RNA versus DNA.
I know
I
DNA
wonder if it's
rules
the same
now.
one that I saw yesterday.
Probably.
DNA rules now in terms of most life forms on Earth are DNA based, but only some viruses have RNA genomes, which we believe is left over from the RNA world, which was first on Earth. And so it's a good molecule.
I'm okay with RNA. That's good.
I want to ask about the fact that they look at whether the responses they got were Th1 or Th2. And with the RNA-based vaccine, they got mixed Th1, Th2. And with the protein vaccine, they seem to be skewed more toward Th2.
Right.
And so I wanted to know whether it's better to have that mixed response, or is it that it's better to have the Th1 response?
First, we need Brianne's primer on TH1 versus TH2.
Right.
Okay.
I have to look this up every time. And I recommend, in fact, Wikipedia has a whole article on T cells that includes all this that I have to read every time. But Brianne,
Sure.
make it so that I don't have to look it up anymore.
All right. So any of these types of T cells that have an H in their name are CD4 T cells. They're helper T cells. And those T cells, when they get activated to become a specific type of CD4 helper T cell. They could become TH1, they could become TH2, they could become TH17, TFH, all sorts of things. And the difference between those is what types of cytokines they produce, which may be useful for different types of responses.
Cytokines being small molecule hormone type things that signal cells to do different things, right?
The messenger molecules of the immune system.
Okay.
Basically, some helper T cells will learn to make cytokines, will be differentiated to make cytokines that make them really good at activating macrophages. Others might make cytokines that make them really good at turning on B cells. And those are the different types of TH1, TH2, TH17, T follicular helper, et cetera. TH2s are in some ways, to me, kind of the bane of my existence, because TH2s are very strongly linked to things like allergic responses. They're usually responses to parasites. They're not what we usually think of as being particularly protective against viral infection. TH1s can be quite protective against viral infection because of the cytokines and that they make and the types of cells that they help. So the idea here is that it's good to make a more TH1 type response with these vaccines. And in fact, a TH2 type response might lead to a little bit of pathology in some cases.
So I have it in my head somehow that, I'm going to make sure I got this right, that a response that's skewed towards TH1 is skewed towards B cells, antibodies. Have I got it backwards? And TH2 is skewed towards cellular killer T cells, et cetera. Am I oversimplifying it, or I got it backwards?
You got it a little bit backwards, and that was kind of true when we only knew about TH1 and TH2.
You're an old guy.
When we got some other subsets, it got more complicated.
Okay.
So they look for neutralizing antibody, which is obviously important. And, these immunization with the protein induces antibodies. It induces long-lived plasma cells, but they're not neutralizing. And the mRNA immunization gave good levels of neutralizing antibodies. So it's just, again, you need those germinal centers to make high-quality neutralizing antibodies. It's just remarkable. It couldn't be more clear, right?
Yeah, I like the point that Rich made earlier about using RBD versus using spike. One thing that could be a little bit of a difference here is spike is a really big protein.
Yeah.
And it probably is providing some parts of the protein that can allow for a good T cell response. So the T cells can help B cells really well. Because RBD is shorter, it might be harder for it to give a good T cell response.
Yeah, I think a long time ago, John Udell said it's not a good idea just to use RBD. Probably it's just you're missing a lot of epitopes. That's what he said. Yeah. The mRNA vaccines are also really good at inducing these T follicular helper cells that are SARS-CoV-2 specific. And as you heard earlier, those are important in the germinal center development. And then, yes?
It would have been interesting to compare an mRNA vaccine that was just the receptor binding domain. Because those have been, Pfizer originally
That's right.
Was testing both full-length spike and the receptor binding domain vaccine. Okay.
Yeah.
And they chose to use the full length, I think, because it worked a little bit better. But the receptor binding domain vaccine did work in animal models and et cetera. And so that would be closer to apples to oranges. But
And they do some experiments here with an RBD mRNA vaccine.
They do.
Do they?
Yeah. Yeah.
oh,
Yeah,
sorry,
there are some
missed
in
that.
here. Yep.
Oh yes, I see.
I mean,
yes.
I think most of one of their conclusions is most of the neutralizing antibodies are focused on the RBD, which kind of makes sense, right?
Yeah.
But not all of them.
I mean, it's not. Yeah.
And
It's
even when
not
you
an
give
unreasonable thing.
even when you give the entire spike mRNA, many of the newts are focused on the RBD. They also look at. We talked briefly about the Th1 versus Th2. They also look at, what controls their one versus the other in terms of transcription factors, and again, the mRNA vaccines are better at inducing the right molecules that are needed for this formation of germinal centers. And then they do a second immunization. They do a boost, and they find with the mRNA, that gives you a second, as we mentioned earlier, gives you a second wave of germinal center responses. You get more neutralizing antibody formation. But again, the protein, even with a boost, you don't get very good germinal center formation. You don't get good neutralizing antibody formation. But that's the story. It's a very well done paper. A lot of good work here, even from my non-immunologist eyes. And really, the mRNA vaccines are way better at making germinal centers, at making long-lived plasma cells and B cells and neutralizing antibody. So when people say...
At least in this comparison and in mice.
Yeah, in mice.
Yeah.
So the next thing is to go in people, and we're doing this experiment. We're giving some people mRNA vaccines, and others are going to get protein vaccines. Although, as Rich said, the protein vaccine of Novavax is different. It's coated in a lipid nanoparticle, so it may be different.
Actually, it's not coated in a lipid nanoparticle. It's the full-length protein that's essentially aggregated by its C-terminus. So it's a little, almost looks like a little star.
Right.
It's its own nanoparticle.
That's right.
But, I forgot. Oh, yeah. One of the points they make here is that this basically sets a standard, okay? And that it would be appropriate to do head-to-head comparisons like this of a bunch of different candidates that are out there. Even, I would think that head-to-head comparison with now what are becoming the new gold standard mRNA vaccines, anytime you come up with something new, you probably want to do this kind of comparison, though mice lie.
Mm-hmm. But you could do this experiment in people because these are just vaccines. It's not like you're infecting people. Just give them mRNA vaccine versus a protein and compare this. You can't pull out their lymph nodes and section them and stain them, but you can do,
Right.
you could look at what's in the blood, right? I guess, Brianne, that would be the
Yeah, exactly.
thing you could do.
So, Brianne, help me out here. I want to go back to this conclusion about Th1, Th2, because the protein vaccine is giving us primarily a Th1 response. Is that correct?
That was the other way. The
Other way,
mRNA.
Th2. See, I got this problem. But the mRNA vaccine is giving it's doing both.
Mm-hmm.
Yep.
Yeah.
Yes.
Protein vaccine is doing primarily Th2.
Yes.
And the mRNA vaccine is doing both. Now,
Yes.
somehow that must be significant
Yeah.
Yes.
for the ultimate outcome.
So
Can you elaborate on that, why
Sure.
that is?
So Th1 cells are going to be better at turning on things like CD8 T cells. You're going to see class switch to some antibody types that are useful. And you are less likely to see, and this is based on some other vaccine models, some immunopathology.
So they say, Rich, in the discussion,
So
the
it's
fact
like
that the mRNA vaccines generate mixed Th1, Th2 might be a desirable feature to potentially avoid vaccine-associated respiratory disease, which has been connected to immune responses skewed towards a Th2 profile in other preclinical and clinical studies on different respiratory viruses.
Exactly.
Yeah.
That's a much fancier way of what I meant by immunopathology.
Yeah. I just wanted to, because that's
but that's really important, because this idea of disease enhancement is something that we've been talking about constantly since the get-go.
so that
this
is
a really nice roadmap for how to compare vaccines going forward. And the thing that I wanted to point out is that it's starting to be a recurring theme that people are putting a section in their discussion called limitations of the study.
Yeah.
This is, people would in the past perhaps, put it in there, but they wouldn't necessarily call it out with a bold heading.
Yeah.
And I think that's really helpful.
Yeah, I was going to comment on that. I agree, Kathy. As a matter of fact, as I was reading this paper, I was anticipating that they might do that, because I've become accustomed to it now. And I was pleased when I saw that it was there, because that's, I'm thinking of, well, what about, the whole time I'm reading a paper, what about this? What about that? And I'm thinking, well, maybe they'll come up with that in limitations. And sure enough, they did. It's good.
By the way, Kathy, they have this sentence here, which I think you'll like. They write, at least 2 reasons could explain the superior ability of SARS-CoV-2 mRNA vaccines.
Yeah, I had to look for a while to find the second one too, but yeah.
The ability to do what, folks? I'm sure they'll.
Yeah.
And this is a pre-proof, so maybe they're going to proof it, right?
Yeah, that's the idea.
Right.
Anyway,
there were a couple of places where they questioned or interrogated, and I thought, couldn't you just examine?
Yes.
So I keep coming back. I keep coming back to respiratory syncytial virus.
And that's the one that they referenced.
That vaccine that caused an enhanced disease and was withdrawn after being distributed. What was that vaccine? What was what was the platform?
It was a formalin inactivated whole virus particle vaccine.
So that would be more like a protein vaccine, right?
Well, it's got lipid in there too, right?
We're all nodding here. We don't know.
It's the whole particle. It wouldn't be just a protein base. It's a whole particle, right? With lipids and nucleic acids inside.
But it's not replicated.
No, it's formalin inactivated.
So there's no on-site synthesis of antigen or anything like that.
So that induced a Th2 skewed response. And that was a problem.
So what I'm really looking forward to, I'm wondering whether some of these new platforms, like the mRNA platform, and we looked just last week at the Moderna site, and they have an RSV vaccine in the pipeline.
Yeah.
Okay. I'll be interested to see whether these new platforms solve some of those old problems.
We learned yesterday from Sally that they have a CMV mRNA platform. They're doing a couple of different viral proteins in that one, not just one. Mm-hmm. That could be very exciting.
Very exciting. I think this is potentially a revolution in vaccinology, this whole event, the pandemic.
Could be.
It's great.
So the reason I read that sentence is because why does the mRNA vaccine induce superior germinal center formation, antibody formation, et cetera? So they say maybe one is because the protein is translated directly in the antigen-presenting cells, right? And that may allow for more continuous presence of antigens that are displayed on the cell surface, right? Maybe better than a recombinant protein. You only have a certain amount that you put in, and then, so that makes sense to me. Mm-hmm.
But now, isn't the presentation of antigens different when they're initially delivered as a protein versus something that's made in the antigen presenting cells?
Yes.
Okay. And so that to me is fundamental to this whole thing. It's MHC1 versus MHC2. Am I talking right?
How it gets onto the MHC1 and MHC2.
Okay.
Yeah, the protein has to be taken up and digested, right, Brianne?
Pardon?
The protein has to be endocytosed, processed, and then displayed.
Yes.
And the mRNA is made endogenously, like a viral infection would make the antigen, right?
There's a little bit of question about whether that mRNA is made and the cell dies and is endocytosed.
Yeah.
As well. But yes, there would be differences in exactly how similar to the viral infection and viral protein production in the cell. Those two pathways are doing.
Okay. Very cool. Check it out. Good stuff.
Actually,
really good.
People can find this paper, right? It's
They can find it.
right?
They can go to the show notes, microbe.tv/TWiV. You'll find a link to it, and you'll go directly to the website. And there you could...
if you just look at the first figure,
it's okay.
It has the pictures of the germinal centers.
Yeah, it's okay.
And it's really quite dramatic.
It's just a little... Yeah, it's okay. The ramps are not great, I think, but it's okay. All right.
No, I'm not talking about the graphic thing. I'm talking about the first data figure.
Okay.
Figure 1D has the actual micrographs of the germinal centers.
I don't like the graphic abstract so much. But.
No, that's not.
I agree.
All right, let's do a couple of email.
No, let's do 6 more papers.
Yeah. Kathy, you have a follow-up?
Yeah, because we did the TWiV last week with some people from Michigan, I emailed them as sort of a heads up. And so I struck up a conversation with Jason Knight, who I'd not met. And he said, oh, yeah, my wife listens and she's not a scientist. So he listened afterward. And I had also told him to watch the one with Kate. And he liked our discussion of his paper. This was about the autoimmune antibodies to phospholipids and so on. And he said, yeah, so you did a good job on that. And between that and space, you guys are just killing it. So that was kind of fun. And then it turns out he said something about, he was lamenting that we had not called out rheumatology, because he said we're now destined to just continue to be in the backwaters. So when I tweeted something about it, I tried to include rheumatology. And he said, and by the way, I'm secretly a virologist. He trained with Earl Robertson and worked on Epstein-Barr virus. And so he did an MD PhD and started his PhD here with Earl, and then Earl moved to UPenn, and then Jason came back and finished his MD here. So we misspoke, but we didn't know in saying that these people are not virologists.
Okay, cool. All right, we have the first from Leilani. Hello, Vincent and TWiV crew. Would you please discuss this article? I'd really appreciate your review and thoughts of it. Also, could you explain it in layperson's terms as much as possible? So the link Leilani provided broke since the first time I saw this, but Kathy has what may be the right link. Right, declining prevalence of antibody positivity to SARS-CoV-2, a community study of 365,000 adults. And this was done in the UK. This is an interesting study of prevalence of antibodies to SARS-CoV-2 using a self-administered lateral flow immunoassay, very much like this one that I have here in my hand, which was provided to me and which I administered to myself or on myself. And this assay is made by Fortress Diagnostics. So I found that. And you cannot find what it's looking for from the website, but it's a lateral flow assay that you can do at home, basically. And so that's why they could get 365,000 adults. I think it's the whole population of England, right? Just kidding. Just kidding. Okay. And then they did some comparison of the results that you would report with laboratory neutralization activity. And so here, oh my gosh, this font is so small, I can barely read it. Let me blow it up so I can tell you what the finding. So basically, 17,000 positive tests over the 3 rounds of study in this test. Antibody prevalence, which means how many people in the population have antibodies against SARS-CoV-2, declined from 6% to 4.8% and 4.4% over the 3 months of the study. And Brianne will tell you that's not unusual. If you're infected, you make antibodies, and they slowly decline, right? You don't need to keep them at high levels forever, but they will not go to zero.
You don't want those lymph nodes to stay huge forever.
No, not at all. And So that's normal. There was no change in antibody
is that
positivity.
like a 5% seroprevalence?
Well, it was 6 initially and then went down to 4.4%.
Right. Okay, fine. Yeah.
In all age groups, smallest overall decline in positivity in the youngest age group, 18 to 24 years. And the largest decline in the oldest group. Does that make sense, Priyan?
Old guys just can't hang on.
Yeah, I think that makes sense based on what we know. The link that Kathy provided also links out to a preprint that shows the data on this. And we talked about a couple of papers with antibody longevity data previously.
Right.
And the data here look pretty similar to what was seen in those studies. So this looks like a typical immune response sort of contracting as you go down.
Now the problem here is they say these findings provide evidence of variable waning in antibody positivity, such that at the start of the second wave, only 4.4% of adults had detectable antibodies. But using this lateral flow test, okay, it may not be as sensitive as a laboratory ELISA. Okay, so there may be enough antibody there to protect you from infection or from disease, right? That's the key here. So these lateral flow assays are not meant to tell you the lowest amount of antibodies you have. They're supposed to be a screen of the population. How many people have been infected? So I think the waning part is not really good to do using these lateral flow assays. It's more of a qualitative view of the population, I would think. But
plus, I'm getting a sense that this, the whole pitch here seems to be that this is bad.
Yes. They say antibodies are going down, but
and that's not my conclusion.
No.
It sounds to me, in particular, in combination with, although it was a shorter time scale, but in combination with our discussion with Shane last week, that this is normal, right?
Yeah, it's supposed to go down. It's supposed to go down. And it may be 0 on a lateral flow, but you're still not 0, right? That's the point I'm trying to make. Okay? Because the sensitivity of a lateral flow assay, one of these critters here, is not great. But you may have enough antibody. So, they're saying maybe this is bad, but not necessarily.
Right. I also find it interesting that the seroprevalence in this study with this assay, et cetera, all those qualifications, is nevertheless significantly less than 10% in a country that's had their share of problems with this pandemic. So anybody out there who thinks we're turning the corner on this is wrong.
Kathy, can you take the next one?
Yes, Lauren writes, greetings from Seattle, where it is a Monday in November and therefore cloudy and 7 degrees Celsius or 45 Fahrenheit. After listening yesterday to your conversation with Dr. Shane Crotty in TWiV 684, I thought I might have something worth sharing. Though I am a mere architect with a physics degree, I've been an avid TWiV listener since early March, understanding maybe 80% of each episode. Over the summer months, I wish there was something I could do to contribute to making this pandemic less pandemicky. So when the large-scale vaccine trial started, I raised my hand to volunteer. I am now in the Moderna phase 3 trial and just had a 1-month in-office health check with the study after getting my second shot in late October. They took 8 tubes of blood today, and the study calls for similar checks at 7, 13, and 25 months for all 30,000 of us. I asked how the blood I was giving them would be tested, and they said it would be checked for virus, antibodies, and more. When I asked, they confirmed that the goal was to not only catch asymptomatic cases, but also determine if the vaccine is protecting against infection. For those of us who actually show up for the 7, 13, and 25-month checks, they should also be able to chart how our response is declining, assuming it will. Is there anything else you want them to check? The logistics are complicated, but they are doing it anyway. By the way, I am pretty sure I am a control since I had essentially no reaction to either of the shots. Then again, maybe I am wrong / lucky. Lauren.
You need to, if you're in these clearly these mRNA vaccines are going to be stimulating your germinal center. You can go back to the doctor and say, man, my germinal centers are really pumped up.
Yeah. Yeah. If you're going to, these are in the muscle, the arm muscle here. So your lymph nodes in the armpit or the axilla are going to be swollen. You could easily do that. You could feel it every day after you get the shot. Check it out.
Actually, it's axillary lymphadenopathy, right?
Yeah.
It is.
Yeah, certainly not your foot. But
I like that he asked us if there's anything else we want.
Yeah, that's a good question. What else should they check besides? So they're going to look for virus. I'm not sure virus is a great thing. They should look for it for sure. But viremia is quite low, and I'm not going to sure what that is going to tell you. But certainly antibody, because if you see a spike later on, you may be getting infected in the absence of disease. So that would be good. Brianne, lymphocytes?
Yes, I would certainly want to look at lymphocyte numbers. Obviously I can imagine all sorts of fun T-cell assays one could do.
But you would want to know if there are virus-specific lymphocytes, right?
Exactly.
Both
yes.
B and T cells, correct?
Both B and T cells.
And you could look for memory B and T cells also?
You could look at some flow cytometry markers that give you some hints about memory.
I mean, that's what Sean's study was doing last week, right? They had just blood draws and they were looking at antibody, memory B, and memory T cells, right? So that's what would be great to look at. Wow, look at all those tubes, 30,000, and there are 8 tubes, 4 different time points. Wow, that's a lot of tubes.
And don't a lot of those assays have to be done with fresh material? Like you can't freeze this stuff and come back to it later?
There are some that can be done on frozen cells.
Okay.
I'd like them to take, I wonder if they do, take a saliva sample. Because if you wanted to look for virus, that would be the place to look. Because I want to know whether after you've been immunized, you can be still reinfected, say, asymptomatically. Sure. I think that's a really important piece of data.
I agree.
And my understanding is that saliva is a perfectly good source of virus and that it keeps.
Now,
at least on the short term. I don't know if you can freeze it. Well, certainly you ought to be able to freeze it and get RNA out of it later on.
I think so. I was on some call recently where someone said you really need a mill of saliva, and because of the sensitivity issues, I think a nasal swab would probably be better, and it's a lot easier to do. Just give people a little swab, boom, boom, boom. That's a scientific term, by the way. Boom, boom, boom.
Yeah.
I don't know. I thought the saliva test was pretty easy too.
A mill is not easy to cough up, depending on.
Oh, I can come up with a mill.
Yeah.
Well, in this call, there were docs who said it was an issue. So I'm just repeating what they said.
Well,
fine.
A oral trick that we know is when you're on stage and you feel a tickle in your throat and you don't want to have to cough, is to bite your tongue and create some saliva so that you have something to swallow.
Wow, great idea. I'm doing it. It worked.
Yeah, it worked.
There you go.
I didn't even know that, Kathy. Good one.
Cool.
I like that.
The other one is if you have to cough or you feel like you're going to have to cough, is don't look down. Instead, look up, because then that kind of stretches things out and you might not have to cough. So
I just push my mute button. I just push my mute button like this, right? And you can't hear me anymore. Oh, but that's a great idea. I've been in many situations where I'm about to cough. There's no water left. The cup is empty. Good trick. Okay, Rich, you get the next one.
Jim?
Yep.
Jim writes, Dear TWiV team, as a faithful mask wearer, love to get your take on this article in the Wall Street Journal. Love the show. Thanks. I have not looked at this.
I did.
Okay, Kathy, what's up?
It's an article in the Wall Street Journal by Joseph A. Ladapo, who is an MD, PhD in health policy research, cost effectiveness of diagnostic technologies, and population burden of cardiovascular disease. And his point is that at first we were told not to wear masks, and then almost overnight the recommendations flipped because, as he says, the risk of asymptomatic transmission And he claims that this is a weak rationale, given that pre-symptomatic spread of respiratory viruses isn't a novel phenomenon in public health. Asymptomatic cases of influenza occur in up to a third of patients. And no one has called for mask mandates during flu season. So I found that argument not to be very strong. And then he says, by paying outsized and scientifically unjustified attention to masking, mask mandates have the unintended consequence of delaying public acceptance of the unavoidable truth. In countries with active community transmission and no herd immunity, nothing short of inhumane lockdowns can stop the spread of COVID-19. So the most sensible and sustainable path forward is to learn to live with the virus.
It's just wrong.
So it's basically a let it rip through and masks don't work. And so there was an article that I also put in, what scientists say about masks, basically wear one. And this was in the Science Times yesterday, Tuesday. But then I found the online version, and the online version has a whole section that was omitted from the paper copy. And basically, they talk about how it's harder to study masks than drugs or vaccine. It's really hard to do randomized clinical trials.
Yeah.
It's particularly difficult to assess a mask's benefit to the wearer because you have to be able to measure what's behind the mask and what's getting through the mask into their mouth.
Yep.
And so I think there is plenty of evidence that masks are useful for the larger population, but they're also useful for the wearer. And so this article in the Wall Street Journal, I was very upset with.
Yeah, I would agree that it's very hard to test them. And I think most of the studies that have been published are flawed. In my view, the best results come from countries that wear masks all the time, like China, Taiwan, South Korea, Singapore, and they can control infections by doing so. And
the problem is that it's hard to do appropriately controlled studies with a population, right?
Yeah.
You know, just masks, just distancing, or just hand washing, or combinations of the two. But there's absolutely no question at all. But the combination of these things has a profound effect. It's the most important thing we know of so far in controlling the disease burden.
And as I have said before, folks, if you think masks don't work, the next time you have surgery, tell the people in the surgical suite not to wear masks, and you see what happens. Are you serious? Masks don't work? It's not. The masks are not just holding bacteria back. By the way, it's not bacteria, but it's in droplets. It's droplets that the masks hold back. And so a bacteria full droplet, a virus full droplet, either one, the mask works. Why do they wear them in surgical suites? Stop saying masks don't work. It's absurd.
Right. If anything, I would say from some of what he says here, if I try to be generous, because this article makes me rather angry as well. The one thing I would say is, oh, you're right. We probably should wear masks during flu season more often. And we probably should be wearing masks more often to prevent some of these other things that you mentioned.
Yeah, we should. And also when people wear masks, they don't wear them properly for the most part. I see so many people with the mask, below the nose or on the chin. What is that about, folks? Wear them properly. Did you hear your senator, Rich, got mad at someone who told someone else in the Senate to wear a mask? He said
this is Ted Cruz.
Yeah, it's horrible.
It's disgusting. So this was the Wall Street Journal, right?
Yeah.
Mm-hmm. Mm-hmm.
Of course.
Well, why do you say that,
We
Rich?
ought to have some sort of TWiV award, for, just really bogus articles and reporting and et cetera. This really is infuriating.
There are many examples of gatherings of people who were not wearing masks, and there was a very good transmission. And we have talked about CDC reports of situations where people are wearing masks and there are no transmissions. I think that's about as good as you're going to get Wear a mask.
And the choice is not between, lockdown and no lockdown. Okay. There's a path through this. Okay. Some businesses are more susceptible to the crisis than others, bars in particular, restaurants, gyms, etc. But we have learned over time that you don't have to lock the whole thing down. That was before we knew how it was transmitted, really, before we knew a lot about it, that was not an inappropriate thing to do. But we've learned that you don't have to do that. You can shut down parts. You can, and in combination with these other behaviors, you can mitigate the spread, And you can step in because you don't have to do a complete lockdown. You can step in and financially help out the people who suffer the most if you have the appropriate will to do that. So there is a path through this. It's not black and white.
Yeah, this article takes the position that we either lock down or we let it rip, and that's you just said that's not true. It's not true.
That's a false picture.
Well, and that masks are part of that locking down.
Right.
Brianne, can you take the next one?
Yes. Lincoln writes, "Hi, TWiVers. It's a lovely 22C 71F here in Melbourne." Is that right?
Very good.
Vic, where we are now becoming world renowned for having effectively reduced our cases from over 700 new cases a day, and sadly having said goodbye to 800 of our state's people on account of COVID. Now with a 14-day rolling average of just 2.6 new cases a day, having had 2 days earlier this week at 0 new cases with over 20,000 tests each day from our 5.5 million population. But moving on to the news, I came across this article today that piqued my interest. I know that the idea of copper-laced masks has come up on the show, and I've even seen some copper-laced snoods, kind of somewhere between a scarf and a skivvy neck, being sold as a preventative measure too. But now I'm seeing the idea of using PPE, or as this article proposes, rephrasing it as public health equipment, or PHE, being taken a step further. I'd love to hear your views on it. And he gives a link that I have not yet looked at. His end of his letter is, "Not being a scientist, but a voracious sponge for knowledge, I can see huge value in the mass production of a solution like this presents, if the science and virology checks out. This year has quite frankly sucked, but learning of and becoming quietly infatuated with each new TWiV episode has made it suck just a little less. Thanks and best of luck throughout the remaining months, not of this year, but of this global health crisis, for which your words provide some guidance that I think the whole healthcare and political community would do well to heed, should they not already be." Lincoln.
Well, the article references a paper, a journal article in a journal called Matter, which I can't say I've ever looked at before. It's published by Cell Press. It's called En Masse Chemical Modulation of Respiratory Droplets. And so these researchers have coated masks with materials that would not only concentrate the droplets coming through, but inactivate them. That's the idea. They're polyaniline-coated, non-woven fabrics. And then they show just chemically that this can work, but they don't have any inactivation of any particular virus here in these masks. So it's a really early concept. Seems to me that it might not be a bad idea, but, you know, it depends on cost, on recyclability, and so forth. Whereas, you know, I don't know, a cloth mask you can wash and so forth. So I think copper masks are in development. Michael Schmidt has told us that they're in development, but, you know, where are they? That would certainly inactivate virus infectivity. And certainly putting the right chemicals in a mask would do that well, as long as it's safe. But this is mostly a physical, material science paper looking at how to put things in masks and the sizes of droplets that you can catch and so forth. So there's no virus involved. But I think it's a good idea.
So some time ago, when we were talking about singing masks, Kathy, and et cetera. A friend of mine, a singer friend of mine, turned me on to a singing mask from a friend of his that used a material that basically is charged with some chlorinating compound and can be recharged. And I was very skeptical. I looked this up, but apparently this is the material in play on this mask has been produced for some time by a manufacturer who's into this kind of business. So the idea of a disinfecting type material has been around for some time at least. And I agree. I think that this is, not a crazy idea. I wonder, this would be hard to measure, but how much more effective something like this would be. Okay. If a mask is regular old cloth mask is 90% effective in doing whatever it's going to do, then is 95% more effective going to have that make all that much of a difference in combination with the other mitigation efforts? I don't know, but those are hard to measure. It's not a bad idea. Whatever.
The other thing that's called out in this is the thing about PHE versus PPE, and it's explained. There seems to be quite some confusion about mask wearing, as some people don't think they need personal protection. Perhaps we should call it public health equipment instead of PPE. So, maybe that would help. I don't know.
No, that's a good messaging idea to communicate, even in the name of the device and the behavior, that it's not about you.
Yeah.
I think it's too late, unfortunately. Very early on, we realized that social distancing was incorrect, but people still use that term, even though many people have said, yeah, physical distancing is what it is. We had a sign in the elevator here at Columbia, keep social distance. And I took a marker and crossed it out and put physical. And now all the new signs have physical distancing on it. So,
good.
Nice.
But I still see so many people talking about social distancing because that's the word that was originally used. It's really hard to break it.
Yeah. Yeah. I got our local radio station saying physical.
Good.
But NPR nationally, you'll hear him say social.
Let's do some picks. Brianne, what do you have this week?
I have a book that I recently got. It is called The Types of Scientists Coloring Book. I actually have it right here. And there are pictures of different scientists. All are actually women scientists going through the alphabet, saying things like A is for astronomer and B is for botanist. And it explains what a scientist of that name does and how you would get trained to become such a scientist, and all about what you would do as that scientist, and also has a picture. V is for virologist. And I think it's a really neat way that kids can find out about all of the different things one can do as a scientist.
Does it have meteorologist?
Let's see. What's
I was just asking the other day on our walk how you become a meteorologist.
Actually, M is for microbiologist.
Oh, of course.
Brianne, are you going to color it?
Maybe if I feel very stressed, but not today.
Okay. Do you have things to color it with, like crayons or colored pencils?
I could probably find some colored pencils if I needed to.
Okay. I think that would be fun to watch Brianne color a coloring book. Kathy, what do you have for us?
I have a South Pole video. So my younger brother found this. It's from a channel that he watches. And at the very end of the video, there's kind of an advertisement for that channel or platform. But we have been seeing slides and having slideshows from my older brother since around 1977. And so it was fun to get his take on it. It's a really well done video. And so I think it's worth taking a look at. And then Bill said it's pretty much accurate, although they, for instance, sometimes will take a still shot and pan across it as if they're filming a a video. And they talk about the geodesic dome, and they show some pictures of that. And that's where Bill went the first time that he wintered over. And then the newer facility is why he went back and was involved in the sort of punch list engineering checkoffs of things while that was being finished and so forth. And they show photos of a camp with a lot of tents. That's actually a tourist camp for those people that just fly, ski, or walk in that aren't part of the science research group. There's a couple other things of pictures of emergency generators, but they're not from the pole, which, as he says, this detail matters only to people who have been there and worked on them. So being an engineer, he could know that. And then I had been wondering about what was the situation with respect to the pandemic and people going in this November, which is the beginning of the summer. Basically, they're quarantined for several days in San Francisco before they get flown to Christchurch on a charter flight, where they were then quarantined at one hotel for 2 weeks and then moved to another hotel chartered by the Antarctic program until their flights south could happen. The group of South Pole folks that he's following got to McMurdo, which is on the edge of the Antarctic continent on October 29th. The first flight to Pole wasn't until the 18th of November, which is the second latest first flight ever. There were only about 12 passengers aboard, and the subsequent flights, as of his writing this a couple of days ago, had been canceled due to weather until next week. So it's slowly ramping up, but that's what their precautions are.
PAX is the abbreviation for passenger? I didn't know that. PAX. I looked it up.
CNX is the abbreviation for cancellation.
What is that, an airline thing?
I guess I learned CNX working in a hotel, but yeah, it probably works for airlines as well. Yeah.
Interesting. DX, RX.
Yeah. There you go.
Rich, what do you have for us?
I'm pretty sure I'm recycling old pics here, but we have new listeners. I finally, after a lot of thought, upgraded from an iPhone 6 to an iPhone 12. And I'm very pleased with my decision. And this motivates me to pick the biography of Steve Jobs by Walter Isaacson, which is just a great read. Walter Isaacson, for those who don't know, he writes a bunch of stuff, but he's largely a biographer and likes to biograph individuals who sort of sit in the overlap between technology and humanities, though I don't know how much humanity Steve Jobs has. At any rate, so he's done, I've read, in addition to this one, Einstein and Benjamin Franklin and Leonardo da Vinci. He's done all of those, but this Jobs biography is quite good.
Yeah, I read that a while ago. It's very good.
I still have it on my bookshelf. Haven't read it yet.
Yep, okay. It's good.
My pick is Screenology, continuing in my picks of science art, science-themed T-shirts and prints by Deb Skloot. And, microscopes, E. coli, mosquitoes, phages. She has a great phage print, phages attacking E. coli, which I bought. It's just gorgeous. It's right here. Yeah, there it is. It's really big. It's like this. It's about 2 feet wide by 1.5 feet high. It's beautiful. It's kind of in a shimmery material. So I met her. There's a tardigrade 2 influenza virus, trypanosome, Geiger counter.
Those Christmas ornaments.
Very nice.
I met Deb a couple of years ago at a meeting at Berkeley where I did a TWiV, and I did a little interview with her. So I'll put a link to that. It's not a TWiV, but it's a little video on my YouTube channel where she talked about her inspiration and so forth for that. So her husband was a postdoc in that. Yeah, Britt Glaunzinger. Thank you, Google.
I guess you're right. I think of her as herpes virologist
me
in
too.
general, not as specifically KSHV. Yeah,
Well,
sorry.
It's okay. She does KHSV too. In fact, on a TWiV we did, she talked about KSHV. So her husband, Deb's husband, was a postdoc there, and so she used to talk to him about viruses and got inspired. And Britt in her office has a couple of Deb's prints on her walls, which you can see in that video. It's right there. See where I said here?
Yep.
Okay, got it.
That's the link.
Got it. Cool.
No listener picks today. All right, that's TWiV 685. Show notes, microbe.tv/TWiV. Questions and comments, TWiV@microbe.tv. If you'd like to support us for the rest of November, go over to parasiteswithoutborders.com. Your donation will be tax deductible. They're a 501(c), and they will match it and give it back to us. And when November is over, which it will be shortly, you can go to microbe.tv/contribute. Kathy Spindler's at the University of Michigan in Ann Arbor. Thanks, Kathy.
Thank you. This is a lot of fun.
Brianne Barker's over at Drew University, BioProf Barker on the Twitter. Thanks, Brianne.
Thanks. It was great to be here. And I'm right now looking at this Etsy shop and all the things I want to get.
It's got good shirts. They're nice. I have a couple.
They are.
Yeah, they're very nice. Rich Condit's an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Actually, we'll be back in a few days. Another TWiV is viral.